my academic background is I was an organic chemist. I had a particular interest in understanding glycan structure. My work has moved into looking at the tumour microenvironment, and in particular interested in the tumour tissue and how that interacts with immune cell interaction. So the title of our ABC-funded project is characterising the glycan shield of the tumour extracellular matrix. So the aim is very simply to understand what are the structures that are present within the extracellular matrix. This is a component of all tissues, and actually it's been associated with tumours for as long as we've described cancers. It's one of the hallmarks of a cancer. Yet we know very little about what makes up a tumour extracellular matrix, or indeed how it interacts with all of the different types of immune cells and other cells that are present within the stroma. Yeah, so if you think of everybody's made up of tissues, and one component of the tissues is the cells, of course, and the extracellular matrix are all of these proteins which are produced, which cells attach to and informed about their environment with. So they're a very, very important part of tissues, just like cells. So yes, so the glycan shield describes a particular set of structures which we hypothesize are present within the extracellular matrix. So glycans are these post-translational modifications that proteins get coated with, and in the extracellular matrix, around 95 or over, maybe 100% of those proteins have some type of glycosylation on their surface. And we think from some of our preliminary data that in disease, in invasive carcinomas, the type of glycan pattern is very, very different. And the type of molecules that we've seen may well be ligands for receptors on immune cells. So they may behave a little bit like checkpoint molecules, which are very important right now in current immunotherapy strategies. The difference would be that these would be present within the whole of the tissue, so it would take up a much larger part of the immune response than just a cell membrane. Yeah, so from our previous work, we identified a composition of the extracellular matrix. We call it a matrix index, and it comes up very highly in triple-negative breast cancer. So for us, this was a good starting point, since we think it's really relevant to this disease. Yeah, so it's a very simple proteomics method. It's the end output is we analyse tissues by mass spectrometry. The difference is how we process those tissues in the first place. So normally people would be perhaps more looking at what's present in the cytoplasm of the cells or the cell membrane. In our approach, we look at the proteins that are present within the tissue. So it's just a different way in which we process the tissue before we analyse it. So the current immunotherapy strategies that are in place for, particularly for the solid cancers, they're really very exciting, very promising, because if you respond very well to one of them, then you will have potentially progression-free survival. That's something you don't see with conventional chemotherapies. The problem is that currently only about somewhere between 10 to 20% of patients will have a good response against an immunotherapy. So what we think is the extracellular matrix that the tumour has surrounding it acts like a barrier, or in the case of the project, a glycan shield, which physically blocks immune cells infiltrating into the tumour tissue. So if we can identify what those proteins and glycans are that are inhibiting tumour infiltration, then we can hopefully improve tumour immunity. And if we do that, then we can get more people responding to immunotherapy. Yeah, so we have a publication this year in a journal called Cancer Discovery, which was called Deconstructing a Human Tumour Microenvironment, and you can read there about where this matrix index was derived from. You can also find out about us from our website. So we have a website called canbuild.org.uk. So there you can look at the types of data that we've generated. If you've got an interest in looking at a particular molecule, you can even mine our data there and see if it associates with some of the work we've done. Yeah, I think it's really important for focusing what we do and making sure that what we're doing is going to be most impactful to patients. So I think it's very important to understand what the funders want to achieve and also what the patients would like to see research, so that we can tailor our work to have the most impact in that area.